All right, well, welcome to Berkshire Green Drinks. This event is organized by Berkshire Environmental Action Team, also known as BEAT. My name is Chelsea Simmons and I'm BEAT's Programs Director. So if you don't already know about BEAT, you can learn much more on our website, thebeatnews.org. That's also just a great way to stay connected. We try to make it a community resource, so you can find a compiled list of environmental events happening locally, regionally, statewide, that other organizations and individuals send us. We also have an environmental jobs board, and we also have the environmental monitor and public notices on there. So those are just notices about upcoming Conservation Commission meetings near you, or really big projects that we should all be keeping an eye on. And we compile all of that in our bi-weekly newsletter, the Beat News, the Beat News, and you can sign up for that on our website, thebeatnews.org. BEAT is a 501c3 nonprofit, so that means all of our funding comes from grants and generous donations. So programs like this one are only possible thanks to those who support BEAT. If you would like to support BEAT, you can do so by going to the donate page on our website. There are many ways you can, and thank you. Next month, our Berkshire Green Drinks Jessica Duffy will be speaking on Wednesday, March 12th. And she's going to talk about two rare butterfly species found here in Berkshire County, the veined white and the West Virginia white. And she's going to talk about their natural history and just reliance on specific plants, but she'll also be talking about just the threats they face, including some parasitoid wasps that were actually introduced to control the invasive cabbage butterfly. Jessica will also discuss just the impacts of invasive garlic mustard on the West Virginia white population, which kind of crowds out its native host plant and might act as an egg sink. And so this actually all ties into some new volunteer opportunities that BEAT is organizing this spring. We'll be asking volunteers to help Jessica survey the study area for these butterflies, and we'll also be organizing garlic mustard pools in the study area. So if you're interested in helping us with that or just learning about this project, definitely attend that Berkshire Green Drinks next month, and we'll be back to our normal hybrid format with the in-person gathering happening at Hot Plate Brewing in Pittsfield. So before I hand it over to Shelby, our speaker this evening, I just want to remind everyone to please stay muted during the presentation. We'll have a Q&A afterward, so feel free to put questions in the chat while Shelby's presenting, and afterwards we'll go through those questions and people can unmute then. With that, I'm going to hand it over to Shelby Perry, the Widlands Ecology Director, I believe is her title with the Northeast Wilderness Trust. But yeah, I'm really excited about this presentation, and thank you so much for being here, Shelby. I'll stop sharing my screen and you can introduce yourself a little bit more.
Thanks so much for having me. I'm excited to be here. I'm gonna go ahead and share my screen now and put the presentation on. All right.
The bars are gone.
Oh, yay! We were having some technical difficulties with my presentation earlier, but hopefully this all looks normal to you. So as Chelsea said, my name is Shelby Perry. I'm the Widlands Ecology Director at Northeast Wilderness Trust based in Montpelier, Vermont, but we work across the region. And I'm here today to talk about myxomycetes, also known as plasmodial slime molds. But I would be remiss if I didn't take a quick opportunity to tell you a little bit about Northeast Wilderness Trust, just because they are the ones paying me right now. So we're the only regional forever wildland trust in the Northeast. We protect land through easement and fee as wilderness, and we currently protect over 93,000 acres across 6 states, including all of New England and New York, except for Rhode Island. We have no properties in Rhode Island right now. We have a 2-person Wildlands Ecology Team, of which I am the director, and you can learn more at anywilderness.trust.org. And I wanted to share that we had an anniversary recently, and we celebrated it with this One of Many campaign, and I advocated for and got slime molds included on the sticker. So we take a more eco-centric approach to conservation, and we think of humans as one of many species that share the landscape, and try to kind of use that approach when we're doing conservation work. And that's where this sticker came from. And that little puddle looking thing with the, well, I'll call them fruiting bodies in it is a slime mold and fruiting bodies. So what are those? Let's talk more about slime molds. I'm going to start here with a real quick little disclaimer. I am a hobbyist, not a professional. Most of what I know about slime molds is self-taught, and knowledge around this area is rapidly evolving. So I'm going to share what I know to the best of my knowledge, but you will have questions that I can't answer, and there are definitely edges to my knowledge, and you'll probably find some of them in the Q&A. So I'm going to talk about the basics of slime molds, their reproduction, and how to identify some common species in our area. And then I look forward to hearing your questions. Okay, so I'm going to start with a little clarification. Slime molds is a big term. It's used to talk about a bunch of different things. 2 of them are myxomycetes and dictyostelids. Myxomycetes are plasmodial slime molds. That's what we're talking about today. These are things you can see with the naked eye. There are many species. One plasmodium gives rise to multiple fruiting bodies. That looks like these you might be familiar with. This is chocolate tube slime. You may have seen this in your local forest. This is the fruiting body. Dictyostelids, those are cellular slime molds or social amoebae. Most of their life is spent as a single cell microscopic in the leaf litter on the forest floor. There are about 60 species described. There are probably a lot more because they're teeny tiny and we're not really good at describing things that we don't see. And many individuals aggregate to form a single fruiting body. Their life cycles are similar but differ in striking ways. And from what I can tell online, they're often conflated or mischaracterized. And so that's something to be on the lookout for. This is what a dictyostelid looks like. It's a tiny, tiny little fruiting body. This is a magnified photo. So we're not going to be talking about those today, but if you go online and you just like put slime molds into YouTube, you might get some things about all these little individual organisms that come together and form a single fruiting body. That is these little guys. But we're going to stick to the ones on the left, the myxomycetes. So myxomycetes, I'm going to just sort of note that I stand on the shoulders of giants. I have learned a lot of this information from people who came before me, including Stephen Stevenson, who is a professor at University of Arkansas and like the US foremost expert on slime molds, author of a few different books here. I've also learned a lot from Sarah Lloyd. She lives in Tasmania, but a lot of the species are sort of everywhere where conditions are correct for them. So she has a lot of the same species in Tasmania that we have here because spores are teeny tiny. And if they get up into the atmosphere, they can travel kind of everywhere. And then this book, Poulin, Meyer, and Bozonnet, Les Myxomycetes, it's in French. It does not exist in English. It's a 2-book set, but it's like the best key to identifying slime molds. So I've used all of these to teach myself what I know, and I've taken a few classes now. But I just wanted to acknowledge these experts who are how I know what I know. Okay, plasmodial slime molds. This is a plasmodium. This is the streaming active phase of a myxomycete. So I'm going to talk quickly about some confusing terminology here. When I say myxomycete, There's that comes from the derivation of that word comes from myxa in Greek, meaning slime, mycetes meaning fungus. So I try to graphically represent that here with this slime followed by this fungus for you. And then mycetozoa is another term that these things are sometimes called. Occasionally it's eumycetozoa. Eumycetozoa is an infraphylum. Since they've started with systematics, this has gotten much more complicated when originally there was just like a kingdom and then a phylum and then a class and then an order, or King Philip came out. Yeah, kingdom, phylum, class, order in our systematics. Now we have some infraphylums. They come below the phylums. Eumycetozoa is from the Greek meaning true fungus animal. And so I tried to graphically represent that for you. And I just chose this little guy. And that is the infraphylum that all of these guys live under. Mycetozoa was once proposed as a different kingdom that these should belong under because they move like an amoeba, like a protist, living animal, but they fruit like a fungi. So they sort of straddle two worlds. They are not fungi, they are not animals, but they are kind of fungus animals. So for a time, they were considered to be in a separate kingdom. Then they became myxomycetes in kingdom Protista. Now they're in myxogastria is sometimes used instead of myxomycetes. This is more recent. Everything I learned was myxomycetes. I went to look it up to update my presentation for you today, and I found that we call them myxogastrids now. But that's a class that includes all of the plasmodial slime molds. Classifications change all the time. This probably will change by the time I give this presentation again. But right now, I'm going to use the term myxomycetes to refer to the whole group of plasmodial slime molds in this presentation. And they're sort of what they fit into kingdoms and the relations between the orders are not super well understood now that we've started doing genetics on these things. It's kind of scrambled some of them. And I'll talk about that a little bit more later, but all that to say, rapidly evolving area of knowledge. And so what I say may be outdated by the time I'm done this presentation, but it was good, at least now. So let's talk about their life cycle. The basic life cycle of myxomycetes starts with a myxocarp. That's the fruiting body. If you're talking about a fungus, this would be the mushroom. This would be the part of it that is the reproductive matter. So these often look like these purple things that I've drawn here. They're on little stems. They have little tops. They kind of look like little balloons. And then out of them comes spores. So the spores are the dust coming out of the tops of those little balloons. And I drew them much magnified just to the right there to sort of show this process. The spores germinate, and after they have germinated, they turn into one of two types of cells, either a myxamoeba, that's sort of a little, not a mobile phase, it's just a germinated spore. It stays on a wet surface, or an amoeboflagellate cell. Those ones have little flagella that come out of the end so they can actually move around. These two things can and do go in and out of a microcyst, which is a dormant phase. That dormant phase can, if conditions are really dry and they can't, when you're a little slimy thing on the forest floor, you need a lot of moisture. When things get really dry, you need a dormant phase to wait out that dry phase before you can move around, because if it's too dry and you need to be a streaming pile of slime on the forest floor, you will just desiccate and die. So you have to go into some sort of a dormant phase. And then they can come back out of those into either form, the amoeboflagellate or the myxamoeba. Then those individual cells will fuse at some point when they run into another one. And when they fuse, eventually their nuclei fuse inside. This is the point in the reproductive cycle where we go from 1N to 2N, if you know what I mean by that. 1N being the genetic material from only one parent in each being, and 2N being the fused genetic material. So this is also called a zygote. This is like a fertilized egg. This zygote is now the beginning of a new genetically unique individual. It will then divide that nucleus into several different nuclei that have the same genetic material in them. And eventually it becomes this one enormous cell that is basically just a big bag of nuclei is really the technical word. And that's what a plasmodium is. You can't always see them, but some of them you can see. I tried to graphically represent them here with this sort of yellow branching goo. That's the plasmodium, but I included a picture to help sort of illustrate that, similar to the white one we saw earlier, because I'm not sure my pictures do it justice. The plasmodium is the animal phase. This thing crawls, it moves across the forest floor, it hunts and eats other beings, including other protists, including decaying organic matter. And then eventually, when conditions are correct, and I should also add, there is a dormant phase that the plasmodium can go into as well. So if conditions are poor for a plasmodium, it can also dry up and go into a dormant phase. But I didn't illustrate it here. And then when conditions are good and it wants to fruit, it does what's called sporulation. And sporulation is when the plasmodium starts to gather and lift up into those fruiting bodies. It wants to usually move those fruiting bodies up somewhat so that the spores can go a little further. They're not just on the forest floor. And here's a picture of what that looks like. So you can still sort of see the veins of slime on that photo, but you can also see that they're starting to collect into fruiting bodies. So that's the full sort of basic life cycle. Then it starts over, and then you're back at those fruiting bodies, which are making and letting out spores. So here's that process for, I think this is a Steminotopsis typhina, which is in the Steminitis group, the chocolate tube slime group. These are all the same organism. The first photo with the white blobs that look kind of like little clouds, those are at the beginning of that sporulation phase. Then when you start to see the little white fingers rising from the substrate, which is rotting wood in this case, that's sporulation a little bit further along. You see them rise up on little stems in the third one. And then in the bottom series, you're seeing those spores mature. And by the last one, you're starting to have more mature spores. They're covered with a coating that will eventually erode away, and that allows the spores to be reduced or released. So this series of images were all taken about a day apart, give or take. So it happens pretty rapidly. We're looking at less than a week to go from the white goo balls to the actual slime mold fruiting bodies you see down in the bottom right. So the active phase, there's different types of that, and different species of slime molds have different kinds of plasmodiums. So what we've seen in all of those pictures, and virtually every plasmodium you ever see, is called a phaneroplasmodium, from phaneros meaning visible. These are all the ones you see. Every time you see that goo going across the veins of slime, across a leaf or a log or something like that, it's always a phaneroplasmodium. Other species have aphanoplasmodiums. This is from Aphanes invisible. They're really small, fine, lacking slime sheath. That's common in that order Steminotales. So that one that I just showed you, you would not have seen that plasmodium streaming across. It would have been small, mostly internal to the log, and it will come out when it's fruiting time. And then there's protoplasmodium, from protos meaning first. These are microscopic. They give rise to a single fruiting body. They're common in the group, in the order Echinostelliales. That's an uncommon one. You don't see them very often. They're small individual fruiting bodies. So they're not commonly seen because they're hard to see, right? We see the ones that are conspicuous much more often. All of these things are very, very small. I'm going to give you something for a little bit of scale in a minute. So those are all the kinds of plasmodiums. There's also different kinds of fruiting bodies, also called myxocarps. So these myxocarps, I tried to draw them, paint them for you here, but I don't think I did them quite justice with my illustrations. So I'm also going to show pictures of each of these. But there's a plasmodiocarp. That's when the plasmodium, kind of that streaming slime phase, just gives rise right to a fruiting body right where it is. And it kind of looks like layers of slime that have kind of hardened into little tubes. So that's this one. There's a sporocarp. This is those individual stalked fruiting bodies, and they usually are separated and each on an individual stalk looking like little balloons. Sometimes they're nodding, sometimes they're flattened or hot dog shaped. And then the next one is a pseudoethalium. The pseudoethalium has, it looks like it's all one big glob, but it's actually a bunch of those little tiny fruiting bodies all stuck together. So if you did a cross section, that drawing that I'm showing there, the right side is a cross section and you see the orange coating the brown spores on the inside. That's the spores are all individually coated by that outer surface. And then the last one is an ethalium, and an ethalium is a solid mass of spores covered by that outer surface. So in that cross section, we'd see that orange outer surface with the brown spores in the middle. These are all really tiny. These photos, you can kind of see in this photo the little sort of bits of moss on the log there. We're looking at, these are sort of, this one in particular is Lycogala epidendrum. It's called wolf's milk slime mold. Probably gets about as big as a dime, maybe. Probably these guys are about the size of smaller than a marble, maybe like a Tic Tac kind of size. So anatomy of a sporocarp. These are the fruiting bodies. There's kind of some standard terminology here. Not all of them have all of these, but I'm going to kind of run through. There's a lot of vocabulary in slime molds, and I'll just share candidly that I learned a lot about these before I ever talked about them out loud to people. And then I presented them for a period of time and realized I was saying most of the words wrong because I'd never said them out loud. So I'm going to try to teach you how to say them correctly today. If you imagine a slime mold fruiting body as a balloon on a string, and the balloon is full of Easter grass, and in the Easter grass is glitter, okay? This is my comparison. So the Easter grass would be the capillicium. That's the curly stuff sticking out of the top of the balloon. That's a fibrous stuff that's inside the top of the fruiting body. The spores are sometimes attached to it, sometimes just loose within it, but it holds them aloft inside that fruiting body. That's the spore mass, what's up in there. That's all the little sort of freckles. The balloon itself would be called the peridium. The peridium is a sometimes shiny, sometimes iridescent coating on the outside of the top of the fruiting body. The sporocyst or sporotheca, that's the top, that's the balloon, the top portion. That's everything, the balloon and everything inside it. Not all of them have a stipe. We saw some that were right on the medium that they're growing on, but some of them have a stipe, just like a mushroom. That's the stem. And then the hypothallus is the surface that sort of attaches the fruiting body to whatever it's growing on. And again, not to scale, right? These are tiny, tiny things. Some of them are really small, so I threw some in here to scale for you. I'm mostly joking because I have no idea how big your screen is, but they're tiny, tiny things. They're like little, some of them are pins, some of them are even smaller than that. So I just wanted to sort of drive that home with these little guys down at the bottom here. Okay, so we're going to talk about some basic types, some classification of myxos. And this is a picture of my dog here to walk us through. Serratiomyxales, lyciales, trichiales, echinostelliales, phycerales. See, I'm getting bad at the words already. And staminatales. So these are the major classification. These are the major orders, taxonomic orders. I'm going to go through these one at a time, but Mia's here to raise an objection, which is to say that Serratiomyxales is often considered to be, or sometimes considered to be within a separate subclass because it forms really different structures from other true slime molds. So again, taxonomy is complicated and ever-changing. So we'll look at those Serratiomyxales first, and I'll show you what I mean by that. I just went through all that vocabulary of what fruiting bodies are and what they look like. Here's the one that breaks the mold. Serratiomyxales. Just to acknowledge quickly, the blue slides with the yellow text, they were borrowed from a PowerPoint shared with me by Steven Stevenson of the University of Arkansas. Rather than just like reword them all and put them on my own slides, I thought I would take it directly and give him full credit. He made these. He's the expert. I'm just repeating it back to you. In Serratiomyxales, the spores are colorless to white, and they're borne on individual stalks. So remember how I said there was a balloon with Easter grass and all the spores were inside the balloon? That's not true in Serratiomyxales. Each one of those spores is its own little thing on a little stalk, and I'll show you a picture in just a moment. Their fruiting bodies are unlike anything else produced in the true myxomycetes, and the plasmodium is watery and not usually visible until fruiting. So it just looks wet and sometimes kind of a little bit shiny or slimy until it starts to kind of raise those fruiting bodies, that the sort of fingers of slime out of the logs. So let's look at one of those. This is coral slime mold, Serratiomyxa fruticulosa. There's sometimes described as two varieties of this, filamentous, which is what we're looking at here. It looks kind of like coral, and poroid, which looks like honeycomb. I think recently those were taken apart into two different species because all of my observations on iNaturalist just got reclassified to Serratiomyxa poroides. But for a long time it was Serratiomyxa fruticulosa ver poroides. So again, taxonomy, ever-changing, kind of annoying. So all of these little stalks have spores on them individually. I'm going to zoom in a little bit here. There's sort of limits to what my camera can do. This is small. This whole fruiting body is probably about the size of a dime or a quarter. So I'm zooming in super tight and see the structures that look like coral. They kind of look like they have like little fuzzies sticking off of them. There's sort of little spots around the edges. It's a little bit hard to see, but each and every one of those little tiny dots on the edges of these filamentous form fruiting bodies are the individual spores born on an individual stalk. They're just like growing out the side. So remember I said there's a poroid form and a filamentous form. Poroid forms here on the left, filamentous on the right. There is, of course, a spectrum because nature does not love our hard lines and simple categories. And so there are very clearly honeycomb looking ones. There are very clearly filamentous looking ones. And then there's a whole bunch of stuff in the middle, of course. One more thing to note, they're often white. They're not always white. I've seen them in yellow and I've seen them in kind of a khaki beige color as well. So this is a yellow one. And what you can see on the bottom that just looks like wet wood, that is the plasmodium come to the surface. That fruiting body hasn't risen yet, but this is the beginning of a larger fruiting. So they often are like fully fruited in the middle and sometimes working on it on the outer edges. Here's another example of that. You can see this is the filamentous form. They're kind of long, skinny things. They're just starting to rise on some of the edges. They're more formed in the middle. And then here's one more look at that same species right early in the rising process. So that plasmodium has just sort of come out of the wood. The fingers of slime are just starting to rise out of the moss. And so this is like, from a distance, you'd look at this and you'd think you're just looking at moss or you're looking at like kind of weird moss. But eventually it's going to look really white and it's going to be all serratiomyx. It's going to be all coral slime mold. I'm going to stop saying these words because sometimes I mess them up. Okay, lyciales. This one has no lime. What do I mean by no lime? Some slime molds contain calcium carbonate. Those of you that are botanists know that calcium carbonate is can be a really important nutrient for plants because it can make other nutrients and minerals more available to them. Slime molds move them around on the landscape. To the extent that it impacts plant populations, I have no idea, and I don't think it's been studied, but I think it's really interesting. Some of them have so much lime in them that they're crusty with white on their fruiting bodies, which I think is interesting to think about. This order has no capolitium, so that Easter grass inside the balloon, There's none of that in this one. It has a protoplasmodium or a phaneroplasmodium, meaning either you can't see it at all or it's the only one you can see. So it depends on the species. And the spores are clear or some shade of brown, yellow, or red, which really doesn't narrow it down very much. Some examples of that: wolf's milk slime mold we talked about a little bit earlier, Lycogala epidendrum. This is the ethalium. Those are a solid mass of spores. This is one of our earliest and most prolific fruiters in the Northeast. You'll see these a lot early in the season, and you'll see them on rotting wood, and they'll look kind of like this. You've maybe seen them already. They start out pink, and then as they mature, they get brown, sometimes gray. If you pop them when they're pink, they ooze out something that's basically the texture of Elmer's glue. Pink Elmer's glue is what it makes me think of. And if you wait until they're fully mature and they've turned brown, they're like little puff balls. When you press on them, they'll shoot out little geysers of spores, usually gray or brown. Another species in this group that's totally different, and one of the more beautiful species, I think, out there, is birdcage slime mold, Cribraria cancellata. This is such a unique little being. They're metallic. They have these really intricate birdcage tops. This one has been cleaned. When you see them around here, often those little bird cages are still full of spores. They have the spores inside there, and the bird cage is sort of what lets them out. There are other similar species that have those delicate cage forms as well, but this one's really common in the Northeast. When you see it, it might look more like this. Sorry about the blurry photos. I've taken a lot of these with my cell phone through like a little magnifying glass. The one on the left, the immature form, I took that right in Hubbard Park here in Montpelier. They fruit in these great big fruitings. There'll be dozens and dozens and dozens of them across a log, but from a distance, they'll just look kind of like it'll just look like a little fuzzy. You can get a little bit of a sense of scale from the leaf of moss that's coming down next to it. That's moss, not a fern. So it's a tiny, tiny thing. And then I collected some of those. So those immature ones that I'm showing on the left, I collected some, brought them home, let them fully mature, put them under a microscope, and that's them on the right. So you can see the bird cage, you can see the spores in there. Trichiales, another one with no lime. It has ornamented capillicium. What does that mean? That means the Easter grass has like little ridges and shapes to it. The plasmodium is somewhere between a proto and a phanero. Again, nature doesn't love our simple categories. Sometimes you can't see it at all. Sometimes it's a great big streaming thing. It depends on the species. Sometimes it depends on the individual. Spores are clear or some shade of brown, yellow, or red, just like the last one. Not great for narrowing these down. Some examples: Trichia decipiens. This is another very common species here, especially early in the year. So as the snow starts to melt, be on the lookout for little red balloons on the end of logs. These are about the size of the top of a pin. If you think of a little sewing pin, the little ends are about that size. They're very, very bright when they're immature. The plasmodium is white or red, sometimes orange. And then these guys are bright, bright red when they're fresh and immature. As they mature, they darken in color to a darker red, eventually getting to this kind of rusty orange brown color at the end there. And that last one you can see all the way over on the right is where the peridium has fully eroded away. We're looking at the capillicium there. That is like somebody popped the balloon and all that's left is the Easter grass. All the spores are born in that, and they'll float out from there. Here's a look at those little shiny immature fruiting bodies. And then in the middle one, you can see on the bottoms of those, the peeling back of the peridium. You can sort of see the little edges of it peeling back. It's like a little vase almost with the fuzz coming out. And then that last photo all the way to the right, they look very fuzzy. In fact, just below those orangey brown ones in the middle, you could see some sort of white looking fuzzy things. Those are fruiting bodies of the same species that got infected or parasitized by a fungus and did not make it all the way to maturity. So they're fuzzy for a different reason. Here's one more look at some of those fruiting bodies. An interesting story about these is that I once I saw stumps beside a trail. A log had fallen across the trail and they had cut it up into smaller pieces to roll it off the trail. And the slime mold had been traveling in the wood. And so where they cut it, on the cut ends of the logs, these little guys were fruiting out in the same place in each section of the tree, meaning the plasmodium must have gone several feet, the length of these logs that were bucked up into, 3 or 4 foot sections. And each end of those sections, the little slime mold was fruiting out of, where it had gone in and where it had come out, which is kind of amazing. Here's Metatrichia vesperium. This one sometimes is called blackberry slime mold or black raspberry slime mold. Here's another view of it. They really can look like blackberries. They have some iridescence, which I'll talk a little bit about later. And then here's one more. This is an immature Arceria species being consumed by a vicious predator, a tiny, tiny snail, which does enjoy eating. Snails and slugs really enjoy eating slime molds. And it's fun to watch too. If you see them eating it, you can watch it go because they're basically see-through. So you can watch them eat it and watch them swallow it and watch it go down their little necks. Here's Arceria obvulata. This is a Arceria species. They have longer, that sporotheca, that top part of the fruiting body, they're really long. They're kind of like hot dog shaped when it's immature. And then when that erodes away, they become these little kind of cheese doodle looking things. And the spores are born in those. All right, Echinostelliales. I'm going to start going fast because I think I'm using up my time. No lime, protoplasmodium. That means you can't see it. Spores white or slightly colored, but not very obvious. Reduced smooth capillicium if present at all. Sometimes they have a little bit of that Easter grass, not always. And it excludes, this group excludes 2 Barbiella and Clastoderma. These are kinds of single fruiting body, teeny tiny slime molds that I'm just not going to cover in here because they break all the rules. So here's an example, Clastoderma deberianum. Oh, that was a mouthful. Individual fruiting bodies on tall stalks. These are each individual beings. And then finally, Phycerales. So lime deposits, these ones are limey. They move around calcium carbonate. Phaneroplasmodium, this is the one that you're going to see moving around out in nature. Spores are dark, black to brown, and the capillicium is smooth, more or less. That Easter grass is like spaghetti. It's smooth all the way along. This is one you might all be familiar with, or at least have heard of, dog vomit slime mold. Probably seen it on your garden mulch. It's Fuligo septica. I wonder if you knew that there's also a separate species called dog sick slime mold, because there's 2 kinds of slime mold that look like vomit. This one's called Mucilago crustacea. These 2 are really common garden dwellers. They live in your garden, they fruit on the mulch, and you can just wash them away with a hose. They don't hurt anything. They don't hurt your beds or your plants at all. If they gross you out, you can wash them away. If they don't gross you out, leave them there and let them live their lives. Another species in this group, this is some bigger ones. This one's called insect egg slime mold, Laocarpus fragilis. Fun fact about this one, it's the first slime mold I found in the field doing work about 10, 12 years ago. And I was like, what the heck is this thing? And that's what got me on this journey of trying to figure out what slime molds are. I saw all of these little guys and I thought, these look like bug eggs, but I cannot think of a bug that lives in our area big enough to lay this many eggs all at once, unless the poor girl's coming back every day for weeks at a time, and somehow they're all the same size and the same maturity. This is not bug eggs. So what is it? It's a worldwide slime mold. It lives all over the place. It fruits on stems or plants, often live trees. It looks like bug eggs. It matures to this sort of dark rusty brown color. Again, like the other ones, that outer peridium will erode away and it'll release its spores. It climbs up trees sometimes. So sometimes you'll see it 2 or 3 feet off the ground to get it in sort of the best place to let its spores go. You see that more often when it's been wet. It's hard to climb up high when you really need to stay slimy. So on wet years, slime molds get a little bit more freedom to go where they want to go. And then again, this picture from left to right is getting more mature. So sometimes it's a bright yellow when it first fruits, and then it'll slowly mature to that dark brownish, rusty brownish red. All right, last one, Stemonitales, no lime. This is chocolate tube slime, for those of you that are familiar. This is another big one. That's supposed to say aphanoplasmodium, I think. I don't know what an aplanoplasmodium is. I think that's typo. Aphanoplasmodium, meaning you can't see it, invisible. It's inside the wood until it's ready to fruit. So it just emerges to the surface and then fruits. The spores are dark, black to brown. You've seen them. They're chocolate colored. And it has a smooth capillation. So that whole chocolate tube on the top, let's look at a picture. Those whole tubes are filled with spores. If you take a little pine needle or twig and you tickle those, you'll get a cloud of chocolate brown spores out of that chocolate tube slime. There's many species. This one's Stemonitis axifera. There are others. The only way you can really tell them apart is with microscopy. If you look at them under a microscope and identify that capillatial network, whether it's a closed net or an open net, how ornamented it is, where the spores are born, that's how you tell them apart. Here's another sort of time lapse. This is one that I did in front of my house over the course of 3 or 4 days. It goes from the white blobs, that's when the plasmodium's just rising out of the woods, and then it of course rained, so in the middle they look rather soggy. That's not their plasmodium, that's rainwater. And then in the last one they've matured and they have those dusty looking spores on the surface. Here's some more pictures of those, just a little bit closer. I'm going really fast now and I'm sorry, but I'm trying to finish on time, folks. Here's another one, just a good look at this one. The spores are mostly out, so you can see that capillatial net, and you can see that stipe goes all the way up through inside those little fruiting bodies. Stemonitopsis typhina, here's another one. They're more individually born. They often co-occur. A lot of times when you find one species, you'll find more. Each of the first 2 pictures has 2 species. The third picture has 3, though it's really hard to see. You've got the little pink guys on the front, you've got the white guys on the top, and then in kind of the back of that log is the chocolate tube slime, all on one log. I'm going to talk quickly about iridescence. So this is Lamproderma muscorum. This is not one that I commonly see around here. This photo is from Sarah Lloyd in Tasmania. It probably exists around here, but I have not found it. It is iridescent and beautiful, like really gorgeous. Not well understood. It's involved many times in numerous different organisms, birds, butterflies, reptiles, octopi. There's a whole range of functions of it, from visual communication to camouflage to thermoregulation, water repellents. In myxos, its function is pretty unclear. It might be layers of waterproof membranes that just refract the light differently, giving that iridescence. But so many of them have it that it may just be a byproduct of those sort of membranes and non-cellular materials building up over time. We don't really know. It's very fun to look at, though. I think they're really pretty. So here's a couple more from Sarah Lloyd's photo collection being iridescent. And then I'm just going to zoom quickly through some slime mold science. So slime mold has been found in amber, over 100 million years old. It's an old organism. You can see those fruiting bodies down in the middle of that amber. Those are in the Stemonitales. That's some real old chocolate tube slime right there. Le Blob in Paris's zoo was an attraction. People came to see their moving slime mold. This is Polycephalum. It's a many-headed slime mold. Slime mold shows us the perks of being a loner. That's a cool picture of sporulation. The emperor's love of slime mold. It's changed how we define intelligence. It can solve problems and solve mazes. It was used to help map dark matter. It has some sort of intelligence without any visible form of brain. We don't really know how it solves problems, but it does. It was used to recreate the Tokyo rail system, which is a highly efficient rail system. They used oak baits in the same shape as the major attractions in Tokyo, and the slime mold quickly organized itself into the most efficient way to visit all of those baits. And that's it. Oh, good. I was racing there to try to get in, and it's 41 minutes on the dot, so I did it. So keep on learning. Here's some of those books that I talked about. Oh, I don't know why the logo's down over there. That was an accident. Ignore that. Check out any of the books. Follow Sarah Lloyd Tasmania on Instagram. You can look up slime mold time lapses on YouTube. It's really cool to watch them go through that fruiting stage. There's a slime mold ID and appreciation group on Facebook for people who are interested. And then I threw support Newt down there on the bottom because wilderness conservation is slime mold conservation. All right, I'm going to stop sharing and answer some questions.
Awesome. That was great, Shelby. Thank you. If anyone has any questions, feel free to raise your hand, your literal hand or your virtual hand. You can put it in the chat. I know we already have at least one. It looks like not two in the chat, so I'll start there. Is the slime mold reproductive method the same as fern reproduction?
Similar but different. Ferns, when their spores germinate, they will live as a free-living gametophyte. And so there's like a tiny, basically sex cell plant. It's as if an egg or a sperm lived alone away from its parent. And then those two will exchange genetic material. Two of those gametophytes will exchange genetic material through a film of water or something like that. And that is how they will get sort of fertilized, germinate, and grow into an adult fern. So that's a little bit of a different process from slime mold. Slime mold's a little bit closer to fungi, the germinating spores, but they do move around, and not some, I guess some species of fungi do. I don't really understand those, so I'm not an expert and should not talk about it. But by and large, fungi kind of stay where they are. And slime molds are like, they move around even in the amoeboid form.
Cool. Okay, we have a few more questions in the chat now. How hard are slime molds to raise in the lab?
Really easy. You can grow them in a moist chamber culture. And so that is like if you took a little, like an agar plate, or even I've done it with like a Ziploc bag that I spritzed it with water. And basically, I read this in the book. I've not tried it, but I've read it in the slime mold book that if you chip a little bit of the outer dead bark off of any tree, and you bring it inside and you keep it in a moist chamber culture long enough, eventually a slime mold will fruit. So that tells you how ubiquitous their spores are, right? They're tiny, they're everywhere, they move far in the wind. So you just need some surface for it to land on and get caught. If you keep it wet long enough, it'll fruit. Sometimes more than one species, sometimes just one. You can also, what I have done is bring home a plasmodium. I found one on a leaf. I put the leaves in a Ziploc, I kept it moist, and eventually a slime mold fruited in there and I could see which one it was. They just need humidity.
Cool. What happens to slime molds in freezing winters?
Dormant phase. They go into either that microcyst phase, or I think the adult dormant phase is called a sclerotium. So they go into one of those two dormant phases. They just sort of shut down. Now, there are a few species of slime molds that specialize in snowbanks and fruit in snowbanks. So there are some that can move around in below freezing temperatures to some extent. I don't know a lot about those species and I have not seen them, but they can move around. Most of our local species are in a dormant phase right now.
And what do slime molds eat?
They eat other slime molds. They eat protists, rotifers, little kind of movie aroundy things in the leaf litter. They also eat like decaying leaves, and they do some decomposition, though mostly they eat decomposers.
Cool. This next question, I'm not entirely sure what it means, but maybe you will. Um, will you be at NAMA, NAMA? Um,
Oh, no. I don't know what NAMA is, but I won't be there. I assume it's a conference. I will be at the Northeast Natural History Conference, though I will not be presenting about slime molds. But I won't be at NAMA.
all right. And John, if you want to unmute or elaborate in the chat on what NAMA is, that would be great. And feel free. Um, Next question. And what else besides snails and slugs eat slime pads?
Slugs and snails eat slime molds. Who else? So there are some bigger ones. The dog vomit slime mold, people eat in South America. Some cultures eat it and they call it caca de luna, moon poop. So there is like a culture around eating slime molds, humans eating slime molds, though I have never tried them and do not recommend or encourage the eating of any organism unless you know thoroughly what it is and what that means. So say that for the lawyers. What else eats them? There are beetles that specialize in eating slime molds. There are beetles that live their whole life cycle in slime molds. And so there are a handful of beetles and other insects that do eat them. They're small, and so all of their predators are mostly small. I think, the big ones are snails and slugs. I bring them home and put them under my microscope, and sometimes like the tiniest little bugs come out of them, and I'm like, wow, who even knew you were in there? So most of their predators are like little bitty things that I couldn't give you names of, but mostly like little bugs. And other slime molds eat other slime molds. They eat each other. Fungi do too. They get infected by fungi and eaten by fungi as well. And vice versa. They also eat fungi. So sometimes you'll see a slime mold covering a mushroom and they will be eating it.
And how can you, I guess, just a question off of that, how can you tell when that's happening, that it's like a slime mold eating a fungi instead of a fungi eating a fungi or whatever?
When it's a slime mold, it will be in the plasmodium phase. So it'll be slime eating
Okay.
a fungi.
Okay.
When a mushroom is eating a mushroom, usually there's like filamentous stuff involved. You can see some of the like either what's the word I'm looking for? Not mycorrhizae, but some of the filamentous fungi. You'll see them, or like a mold growing across the top or something. A lot of times they have like little stalks with little tops. The molds have little fruiting bodies in them. The slime molds, when they're eating the mushroom, will just be like yellow, white, or orange slime kind of netting over the top of the mushroom. Sometimes they will fruit on a mushroom and not be eating it. Then you'll see the fruiting bodies just sort of sitting on top of there. And that's a different thing. So sometimes they climb up things and fruit on top of them. They're not hurting it generally, but some of them will eat them. Again, only those phaneroplasmodiums that you can see. The smaller ones live mostly in the wood and stuff. You wouldn't be able to see them. They wouldn't be able to eat a fungi. They wouldn't be big enough. But the The phaneroplasmodiums, the big visible ones, they'll just make like a network of slime over the top of the whole mushroom, and then it'll be obvious because it'll be real slimy.
All right, cool. That's good to know. So there's a lot of praise and appreciation in the chat, but we have a few more questions. Do you know of any pathogenic spores? And that was my dog just hacking up something, if you heard that. Sorry.
I don't. I don't. I don't. They're so small. A lot of the things that give us reactions for people, like pollen that people have allergic reactions to or have, mostly allergic reactions to, usually have it's like big, kind of ornamented, pointy pollen that irritates us. And slime mold spores are just so small. But I don't know. I'm not really an expert in that. There could be pathogens out there that I'm just not aware of. Everything that I've read about them says if you find them in your garden, they don't hurt anything, don't worry about them. I wouldn't go like breathing in spores on purpose, but I think they're mostly fairly benign as far as I know.
Cool. So NAMA is the North American Mycological Association.
Yes. Okay.
Is a conference and it will be in Southern Vermont this year. Thank you, John. So I think we have time for at least one, maybe two more questions here. Any desert species that only arise when rains come?
Yes, but not well understood. And I don't know them. I know mostly our species up here. There are over nearly 900 species described, and that everywhere I look, I see a different number. There are over 800 species in the key that I have, that Poulin, Meyer, and Bozonnet key. And that's sort of changing all the time as we do genetic analysis. Sarah Lloyd, the woman in Tasmania who I've learned a bunch from, she has discovered new species in her own backyard just by when she finds fruiting bodies, bringing them in, looking at them. So we definitely don't have our arms around the entirety of this group. We don't know what they all are. They definitely exist in places like the desert. They definitely exist in places where they're fruiting on snowbanks. So they are in a variety of conditions. Even I would imagine some common species could live in the desert and fruit when rain happens because they have that dormant phase. They would just need time to just eat enough food to fruit wet, and then they could dry out and they'd be fine. So definitely they're out there. I don't know a lot about them. If you're interested, it's probably an area ripe for research.
Awesome. I think that was all the questions from the chat, and it's just about 7 o'clock. So unless anyone's really itching to ask one last question, I think we'll probably end here. And thank you so much. That was such a fantastic presentation, and I learned a lot. Oh, we have one question, if you don't mind answering one more.
Sure.
Do they interact with leaf miners?
Oh, I don't know, but I would guess no. And I would guess no because they're mostly on decaying stuff. Leaf miners are usually in live leaves, live foliage of things. They eat out a lot of the living tissue before a leaf totally dies. So I would guess that they probably don't interact a lot with slime molds because the slime molds would be in the decaying leaf matter. It's possible occasionally they bump into each other and say hi, but I don't think they have a real overlap in any meaningful way.
That makes sense. Well, awesome. Thank you so much, Shelby, and thank you to everyone who joined us this evening.
Yeah, thanks for having me, and thank you all for coming.